Hello everyone, welcome back to my channel Score Points. In the previous videos, we have learned about pollination. In this video, we're going to learn about outbreeding devices, pollen-pistil interaction, and double fertilization. Outbreeding devices. Majority of the flowering plants produce hermaphrodite flowers, and pollen grains are likely to come in contact with the stigma of the same flower. Continued self-pollination results in inbreeding depression. Flowering plants have developed many devices to discourage self-pollination and to encourage cross-pollination. In some species, pollen release and stigma receptivity are not synchronized. Either the pollen is released before the stigma becomes receptive, or stigma becomes receptive much before the release of pollen. In some other species, the anther and stigma are placed at different positions so that the pollen cannot come in contact with the stigma of the same flower. Both these devices prevent autogamy. The third device to prevent inbreeding is self-incompatibility. This is a genetic mechanism and prevents self-pollen from the same flower or other flower of the same plant from fertilizing the ovules by inhibiting pollen germination or pollen tube growth in the pistil. Another device to prevent self-pollination is the production of unisexual flowers. If both male and female flowers are present on the same plant, such as castor and maize, monoecious plants, it prevents autogamy but not geitonogamy. In several species such as papaya, male and female flowers are present on different plants. That is, each plant is either male or female, a dioecious condition. This condition prevents both autogamy and geitonogamy. Pollen-pistil interaction. All the events from pollen deposition on the stigma to the entry of pollen tube into the ovule, altogether it's termed as pollen-pistil interaction. There are two types of pollen grains. One, it's the compatible pollen grain, and the other one, it's the incompatible pollen grain. The right type of pollen grain is called as compatible pollen grain, and the wrong type of pollen grain is called as the incompatible pollen grain. The pistil has the ability to recognize the pollen whether it's of the right type, compatible, or the wrong type, incompatible. If it is of the right type, the pistil will accept the pollen and it will promote the growth of pollen tube and lead to fertilization. If the pollen is of the wrong type, the pistil rejects the pollen by preventing pollen germination on the stigma or pollen tube growth in the style. We have learned that pollen grains are shed at two cells condition, that is vegetative and generative cell condition. In such plants, the generative cells divides and forms the two male gametes during the growth of pollen tube in the stigma. In plants which shed pollen in the three-celled condition, pollen tubes carry the two male gametes from the beginning. The pollen tube, after reaching the ovary, enters the ovule through the micropyle and then enters one of the synergids through the filiform apparatus. Filiform apparatus, it is present at the micropylar part of the synergid. It guides the entry of pollen tubes. The main function of synergids is to guide the entry of pollen tube. If you want to know about more details of the parts of a flower, I have given a link above. Please watch that dissection of a flower video so that you get the clear idea of all the parts of a flower. You can see the pollen tube clearly in that video. Pollen-pistil interaction is a dynamic process. It involves pollen recognition followed by the promotion or inhibition of the pollen grain. Now we can see the longitudinal section of a flower showing the growth of a pollen tube. The pollen deposits on the stigma and then the pollen tube grows till the ovule and the pollen is carried on to the ovule through the pollen tube. If it is a compatible pollen, the pollen grain will germinate on the stigma, then it will produce the pollen tube through one of the germ pore, and the contents of the pollen grain, that is the male gametes, it will move into the pollen tube. The pollen tube will grow through the tissues of the stigma and style and will reach the ovary. Inside the ovary, we know that there are ovules. After the falling of a compatible pollen onto the stigma, the pollen tube will grow through the stigma and style to reach the ovary. It then enters the ovule through the micropyle and reaches the synergids guided by the filiform apparatus. Now let's learn about artificial hybridization. This is a commonly used technique in plant breeding program to obtain desirable characters. Anthers are removed from the bisexual flowers using forceps, and this step is called as emasculation, and it is done before the anther dehisces. The emasculated flower is then covered with a paper bag to prevent contamination from unwanted pollens by the process called as bagging. The two important steps in artificial hybridization is emasculation and bagging. Emasculation is the removal of anther from the bisexual flowers using forceps. When the stigma of the bagged flower attains receptivity, mature pollen grains collected from anthers of the male parent are dusted on the stigma, and the flowers are rebagged and the fruits are allowed to develop. Double fertilization involves two main processes. One is syngamy and the other one is triple fusion. Pollen grains, on reaching synergids, pollen tube releases two male gametes into the cytoplasm of synergids. Among the two male gametes, one male gamete will fuse with the egg nuclei and will form a zygote. This process is called as syngamy. And the other male gamete will fuse with the polar nuclei and it forms primary endosperm nucleus, and this process is called as triple fusion. Both the process together it forms the double fertilization. We have learned that syngamy is the process by which one male gamete fuses with the egg nucleus to form diploid cell called as zygote. One male gamete plus the egg nucleus it forms diploid cell zygote. This process is termed as syngamy. One male gamete fuses with the polar nuclei at the center to produce a triploid primary endosperm nucleus. This is called as triple fusion. So, one male gamete will join with the polar nuclei and it forms the triploid primary endosperm nucleus. This process is termed as triple fusion. As the syngamy and triple fusion takes place simultaneously in the embryo sac, it is termed as double fertilization. The central cell, after the triple fusion, it forms the primary endosperm cell PEC, and this PEC will develop into the endosperm. Zygote will develop into the embryo. Thank you for watching. Please like, share and subscribe. Keep learning and score points with Score Points.